So this trial was needed because there is still a debate in the TAVI literature whether to perform cerebral embolic protection or not. And this class of devices has arisen because there is still a feared complication of stroke within TAVI. Fortunately, it's relatively rare, and the frequency appears to be declining with latest generation devices, but it's still a present phenomenon, and it's a much feared complication because the impact in terms of morbidity and mortality is substantial. So this study was to try and delineate, is there an anatomical substrate that is of greater risk for stroke that we could focus on with regard to these cerebroembolic protection devices. Within the SENTINEL study, so the SENTINEL study was a multi-center randomized study looking at 2-to-1 cerebroembolic protection within TAVI to TAVI alone. And what it did is it reported clinical outcomes, and it found a significant difference in procedural stroke. But what we did in this study is we looked back at data I collected at CT Core Lab in terms of patient anatomy. So we looked at aortic valve calcium, arch calcium, great vessel anatomy, to try and see, is there a particular anatomy that is of greater embolic potential and also of greater stroke risk. We did that in almost the entire population, and then we were able to stratify the stroke according to randomization. So in particular risk groups, putative risk, what was the actual stroke seen in the protected group versus the non-protected group. We were hoping to find signals that were of greater concern for stroke. But in fact, we were unable to identify definitively a group that was of greater risk. And paradoxically, some groups were of equally great risk that we would think would be of low risk. So for instance, if there was little valve calcium, that appeared to be at least of as great risk, if not greater, than patients with severe valve calcium. And a lot of discussion has arisen with regard to why that is. And it's an interplay. It's an interplay between procedural aspects and the patient anatomy. Some people discussed this morning whether the recapturing, with little valve calcium, there's a potential you would need to recapture more. That certainly may be contributory. Increased post-dilatation, we know, is contributory. Increased recaptures also generate more emboli for a self-expanding retrievable device. And these may be relevant, but the bottom line is it's still, even with a detailed anatomical analysis, it's impossible to identify a strata that is of greater risk. So this is where it becomes a personal opinion. In context of both that meta-analysis and this anatomic sub-study of SENTINEL, what we can conclude is it remains difficult to predict patients that have a stroke. And although there may be some signals with regard to such a serious endpoint, we lack the discriminatory value to identify those at greatest risk. So this is where my opinion comes in. I think a lot of analogies can be drawn to the early era of carotid stenting. So if you look back more than 10 years now, when this technology of carotid filter devices were coming into play, there was a lot of discussion about whether we should be selective in terms of which patients to use filters in carotid stenting. And the end result of that is that now in carotid stenting, it's used in everyone. And I think with such a serious complication, if we can demonstrate a very good safety profile then the question comes, why not use it? And that's my opinion. If one is pursuing a stratified approach to cerebroembolic protection, I think one can look at potentially some procedural aspects. So in the field of TAVI, there are some subgroups that we're more likely to do post-dilatation. And those are patients that say they are small valve-in-valve, so we've started to do things like cracking procedures. And at least anecdotally, there seems to be a higher signal for stroke. So I think the stratified camp can look to such anatomies and say, at least those anatomies I should use it. But there needs to be a patient discussion as well, because although the cost remains, and that's something to discuss on a national level for each country's approval process, but the relative cost of these devices is relatively small. If I had the decision of spending $3,000 to protect my brain, whereas the overall procedure cost is $100,000, then for me it would be a relatively easy decision to spend that extra money. We need to demonstrate the safety. And I think it takes larger studies to demonstrate unequivocally that we can prevent stroke, not just periprocedurally, but periprocedurally in a way that separates the curves long term. So I think a lot of confusion has arisen in the small number studies where curves seem to merge later. And I think that isn't a true phenomenon. I think in the physician-initiated studies, both from Ulm and Cedars-Sinai, as well as some other groups, there has been a consistent and sustained difference in stroke from the early procedural stroke. So I think once we demonstrate in larger studies that there is a significant difference, I think there'll be widespread adoption.